Hey, what's up YouTube? So in this video I'm going to cover 2.2 and 2.3 of the IB Biology curriculum. So first question is just draw and label a diagram of the ultrastructure of a E. coli as an example of a prokaryote. So the main items that you want in this diagram is the cytoplasm, the flagella, which is the tail at the end, the nucleoid containing the DNA, which you can just draw. I mean, IB's not picky on how you draw it as long as you have all the stuff there. Plasma membrane is, so basically you have to be careful. The cell wall is on the outside and the plasma membrane is the inner part of it. And then the ribosomes are just the dots on the inside. And the pili are those hair-like fibers that protrude out of the cell wall. And that's basically your prokaryotic cell. In terms of functions, cell wall forms a protective outer layer and it prevents internal bursting. If the pressure is too high inside, then it will prevent the cell from bursting. The plasma membrane controls the entry and exit of cells. It also has pumps, which allows for active transport. Cytoplasm just contains the enzymes that catalyze chemical reactions of metabolism, and it also contains DNA in the nucleoid. The pili, it basically has an adhesive property to it, which allows it to attach to other prokaryotic cells for whatever function it needs to do. Flagella, they can rotate and propel and essentially cause the cell to move. Ribosomes are the site of protein synthesis, and the nucleoid contains the DNA, which, as we all know, is the genetic information of a cell. So those are the main functions that you probably want to hit. In terms of 2.2.3, the electron micrographs, I never really bothered with those. I mean, probably on the final there's just going to be like 1 or 2 questions on it, so I don't really get too worried about those. And then 2.2.4 is very simple, state that prokaryotic cells divide by binary fission. So basically binary fission, all it is a method of asexual reproduction and involves the splitting of the parent organism into 2 separate organisms. But you don't need to know that. All you need to know is it divides by binary fission. So let's go to 2.3, eukaryotic cells. Basically this is found in animal cells, plant cells, and so you have I start off with the plasma membrane always, and then you can do your nucleus. Your nucleolus is the dense structure inside the nucleus. Your rough ERs basically around the nucleus with its studded ribosomes. Your smooth ERs on the outside of the rough ER. Your Golgi is like a stack of pancakes. Your ribosomes are just the dots, and your mitochondria is there, and that's basically all you need to do. You can put a vacuole if you want. I don't think it's significant. And then the cytoplasm is just the general space. In terms of functions, your nucleus contains the nucleolus and DNA. Your nucleolus is involved in ribosome synthesis. Your rough ER is the site of protein synthesis. It also is a site, basically because the ribosomes are on the rough ER, so technically the ribosome is the site of protein synthesis, and the rough ER is the site of modification of proteins. The smooth ER, basically it contains enzymes that help for a variety of functions. I just chose one, which is it helps detoxify drugs in the liver. The ribosome is a site of protein synthesis. The lysosome contains hydrolytic enzymes needed for digestion. Mitochondria basically produces ATP and is responsible for cellular respiration. Cytoplasm contains organelles. Plasma membrane, same function as before. It's a barrier for molecules, regulates the passage and entry-exit of molecules. Golgi body modifies and packages proteins to lysosomes and vesicles. 2.3.3, I just kind of put a check mark next to it. It's not really significant, just the electron micrographs. So when we're comparing, we always want to have similarities, we always want to have differences. So I started off with the differences. So basically in a prokaryotic cell, you have DNA, which is in ring form without protein, and in a eukaryotic cell, DNA is with protein as chromatin. Now to be honest, I have no idea what it's saying, I just wrote it down. That probably wouldn't be my first choice, but there are some really easy similarities and differences. One is DNA is free in the cytoplasm, in the nucleoid in a prokaryotic cell, whereas in a eukaryotic cell, the DNA is enclosed within a nucleus. In a prokaryotic cell, there's no mitochondria. In a eukaryotic cell, there is. The size of ribosomes are different for each, so the ribosomes are smaller in a prokaryotic cell, 70S, which is basically the unit, and then eukaryotic cell, it's 80S. And also the general size of the cell, it's less than 10 micrometers. A prokaryotic cell, sorry, is less than 10 micrometers, and a eukaryotic cell is usually greater than 10 micrometers. Some similarities are both carry out all the functions of life, because both are cells, both have DNA present within them, and both have some form of outside boundary that contains a plasma membrane. You can also just say both have ribosomes. For 2.3.5, it says state 3 differences. I just chose the easiest 3. Basically, there's chloroplast in plants. There is no chloroplast in animals. The cell wall in plants gives the cell a very rigid shape. There's no cell wall in animals, leaving it very flexible. Carbohydrates are stored as starch in plants, and they're stored as glycogen in animals. Keyword here you guys want to use is like whereas, so that you're really highlighting the difference between them. For 2.3.6, outline 2 roles of extracellular components. Basically, all you have to say is animal cells contain glycoproteins in their extracellular matrix. That's part of the mitochondria. And it's involved in support, movement, and adhesion of cell. So those are the 3 words you want. For plant cells, the cell wall provides the plant with a rigid shape. It prevents excessive water uptake, and it gives plants the structure that they have. Essentially gives them the force to counter gravity. And that's basically all you need to know for 2.2 and 2.3. Hope this was helpful. Thanks for watching.